These videos have been produced using content I have compiled into several ebooks, as you can see here. You can get these ebooks for free. If you are interested in getting your own copy emailed to you, click on the link below, fill out the form and it will be sent to you as soon as it is ready. Good luck in your exams.
Hello and welcome back to Part 2, CSEC Bio Paper 2, January 2017. And in this video, we're covering questions 4 to 6. These will be our structured essay questions, right? So let's get started. So here we are, January 2017, question 4. Part A. We're asked to describe how oxygen in the atmosphere becomes available to human blood for delivery to the cells and explain how it differs. So there are three things going on here, right? Three processes that we're looking at. Firstly, we're looking at this part here, oxygen in the atmosphere available to human blood. So that would be your respiratory system. And breathing, right? Okay. And the next part looks at the way in which plants, so we're going to compare plants. Right? Okay. Excuse me. So how do we go about this? Well, firstly, we have, now how I've put it together is I've broken it up into short paragraphs just to show where the marks can go, right? So we have here the opening sentence. And just as what, you would have learned in English, Miss would have, when you're talking about doing an essay, you have an opening sentence. So here's your opening sentence, right? The summary sentence. Oxygen gets to the blood via the breathing process carried out by the respiratory system. So you summarize the main points immediately, because it is basically a short essay that you're writing, right? So you do your opening sentence, summarize, and that can get you a mark. Then we move into the other parts where we're seeing that now we're going to talk about the breathing process itself, how it works. So for you to get oxygen to the blood, you have to get air into the lungs, right? So air moves into the lungs via trachea during inhalation. So you're describing the pathway that the air is moving. What is the path from the atmosphere to the lungs? And how do we do that? How do we get this air to move from the atmosphere into your lungs? And that is where you will describe the process of inhalation. What does it involve? We have the various parts of the respiratory system that is active during this process. First one is the chest cavity is increased. So that essentially is what you're doing, right? You are increasing the volume of the chest cavity. How are we doing that? We're going to do that via contraction of diaphragm and intercostal muscles. And once you do that, that would lower the air pressure and that would cause the air to move into the lungs from outside inwards, right? Now you can give some details here describing the action of the diaphragm. So you can say that the diaphragm is lowered, right? And the intercostal muscles, they move the ribs up and outward. Right, so that's another detail you can add. There's the contraction of the diaphragm, which lowers the diaphragm. Intercostal muscles, that contraction moves the ribs up and outward. This would increase the volume of the chest cavity. And even with lowering the air pressure, you can also indicate that the air pressure is now lower than the surrounding atmosphere, right? Right? And because of that, the air is going to move from the atmosphere into your lungs, down that air pressure gradient. So just like diffusion, where you have a concentration gradient, and the air and the particles move from high to low concentration, the same kind of process is happening here. Well, I should say a similar process is happening here, where the air is moving from high air pressure to low air pressure. All right. Now we're looking at, so far the air has gotten from the outside into the lungs, but we still haven't talked about getting the oxygen in the air from the air into the blood. So this is where we talk about the alveoli. So now that it's in the lungs, we describe the pathway of the air from the, once it's in the lungs, what is the path it takes to the alveoli. So it's passing from through the left and right bronchi, and then into the bronchioles, and then it would get to the alveoli. So that's what we're saying in this sentence here. All right. At the alveoli now, that is when the oxygen is going to get into the blood capillaries. So it diffuses into the nearby blood capillaries, and it's oxygenating the blood passing through them. And that is how the oxygen gets into the blood. Now, I added a little extra detail in talking about the blood goes to the heart, pump to the rest of the body via aorta, right? But on second thought, I wouldn't give this a mark because it's just asking you how it becomes available to human blood for delivery to the cells. So the part of the delivery of the cells, that part wouldn't really get a mark, right? You can add it as a little extra detail, but again, again, we want to be strict. We want to be miserly with our time. So treat this as a little teaching, a little teaching detail. So now that we've covered what's happening here, in fact, because I took that mark off, I would add a mark here to talk about the diaphragm and intercostal muscles that you mentioned, both of them, and that you mentioned the detail of the diaphragm being lowered and the ribs moving up and outward. That would be that additional mark, right? And then we have the plants now, because we just talked about what's happening in a mammalian system. So how do we now compare that to what's happening in a plant? But remember, it's a kind of comparison question, right? So we have to make some kind of comparison. We have to make some kind of reference. So that opening sentence, once again, you have an opening sentence for your main idea. And in this one, we are saying that we're comparing. So it's saying that, okay, in plants, unlike, and the implied, unlike mammals, unlike us, they don't have a respiratory system and they don't have a transport system with a pump. Now, notice I make sure to emphasize with a pump, right? Because remember, there is a type of a transport system that exists in the plants. You have your xylem and the phloem. However, they do not have a pump like what you'd find in human beings with the heart. So that is the big difference there, right? Then we look at how, so if the plant does not have this respiratory system that does the breathing for it, if it does not have a transport system with a pump, how is it going to get oxygen into? That is when we will indicate that it gets into the plant and the cells via diffusion. So that's the process it depends on. It depends on diffusion, and the pathway is through the stomata of the leaves, right? So that is the pathway. It gets to the plant via diffusion. Also, it gets some oxygen from photosynthesis. Remember, oxygen is a waste product of photosynthesis. So that is where you're going to get some of that oxygen. So instead of the oxygen diffusing out of the plant and being lost forever, the oxygen, some of the oxygen from the photosynthesis is diverted towards respiration and all the other stuff it can be used for. Mainly respiration, right? And those are your 9 marks, right? Remember, we moved this mark up. Right? So we have 9 marks, 1, 2, 3, 4, 5, 6, 7, 8, 9. And there you go. Next part, 6 reasons why humans who smoke heavily have less oxygen available to the cells for respiration. And you're like, 6? How are we coming up with 6? There's more than 6 available. So let's take a look at that. So first one, carbon monoxide. That should be the easiest one to remember. Carbon monoxide in the cigarette smoke, that is going to bind to the hemoglobin and make it unavailable to transport oxygen. So that's the first one. The one that people probably might not make the connection is nicotine. Now, why? Because let's look at the question again. They have less oxygen available to the cells for respiration. So this not only involves the lungs, it also involves the transport system that's going to get the oxygen to your cells. So you don't have to focus exclusively on the lungs alone. You can also focus on the impact on the transport system, because that is what's going to get the oxygen to the cells. So that's why we can talk about nicotine here and the impact on the blood vessels, right? It causes constriction and narrowing of the blood vessels. That limits your blood flow. And of course, that also is going to limit the amount of oxygen that you're inhaling. And that should be inhaled. The amount of oxygen that is transported. Yeah. Typo. All right. So see, even I was thinking hard about the breathing part, but this one definitely has to do with transportation. Transported to the cells, I should also add. Transported to the cells. Okay. Then tar irritates. Well, we know about the tar, right? That's the one we often think about the most as well, because we are talking about irritation, and you all know about the smoker's cough when they're coughing all the time, and so on. So tar irritates the linings of the bronchioles. That leads to inflammation and narrowing of the airways. Right? And of course, that then reduces the volume of air. Now notice that for each of these items, I'm not just only talking about the direct impact the component of cigarette smoke has on whatever organ, whether it's the lungs, whether it's the blood vessels, etc. I'm also talking about how that direct impact in turn impacts oxygen getting to the cells. All right? So you have to talk about both. And yeah, and that's it for tar. And let's look at the others now. So that's 3, right? See, I was able to come up with 4 more. So that's 3 plus 4, 7. So 7, 6 out of 7 options, right? And then we talk about tar again, right? So apart from irritating the airways and causing the airways to constrict, which is what we talked about here, right? The tar also is going to impact or affect the production of mucus. It's going to cause a lot more mucus to be produced, again, hence the smoker's cough, and that is going to narrow the bronchioles even more, which again is going to reduce the amount of air that gets the alveoli. Now, another interesting, right, and the next one is tar again. Tar has a huge impact. The tar is going to basically kill the cilia. So the cilia are those almost like hairs in the linings of the bronchioles that sweeps mucus out, and the mucus which traps the bacteria sweeps that out of the lungs, right? So that's why, especially when you have the cold, you'll notice that you would be coughing, right? And you'll take things to bring up the cold, right? So that's the mucus that's trapping the bacteria in your lungs, and you want to get that out of your lungs, and you cough it out, right? So that would narrow the passageway as well, because what happens, the mucus with the trapped bacteria gets stuck in the lungs. It's not being cleaned out because the cilia basically was destroyed. So that, in addition, would reduce the volume of air getting to the alveoli, because remember, the alveoli is at the end of the bronchioles. So if all of this stuff is happening in the bronchioles, it's narrowing and blocking the passageway to the alveoli. So you could be breathing in deep breaths and everything, but the air isn't getting to the alveoli. It's getting stuck in the bronchioles, right? Then we have tar again and other carcinogens. That's going to lead to lung cancer. Right? The lung cancer is what's going to make breathing difficult. It's going to make breathing painful. And of course, less oxygen is inhaled. Right? Because if breathing is painful and difficult, I'm not going to be able to take a deep breath and bring as much air as possible into my lungs. My breathing is going to be shallow, right? Okay. And then we have the I shouldn't say end game, but definitely one of the latter stages of lung destruction, and that's emphysema, which is essentially a condition that comes from the destruction of the alveoli itself. So all this time we're having problems getting air to the alveoli, but via via various processes, which I did add a little note explaining how it happens. The alveoli themselves are destroyed. Now, you don't have to go into detail about how the alveoli is destroyed, because again, we want to be stingy with our time. You just have to indicate that eventually emphysema occurs, destruction of alveoli occurs, and that, of course, is going to reduce your respiratory surface, because that's your respiratory surface, your alveoli, right? And that's where the gaseous exchange is going. So if the surface where the gaseous exchange is happening is full of holes, that's going to mess everything up. So we have here, so that means you can be breathing deeply and everything, but the air will get to the alveoli, but a lot of it is just going to get leaked out. A lot of it is not going to diffuse into the blood because it's full of holes, right? And that, of course, is going to reduce how much oxygen gets into the blood, and in turn, how much oxygen gets to the rest of your body. So when you see people walking around with their portable oxygen tanks, and they have their face covered with a mask taking in oxygen, they could be patients of emphysema, where their respiratory surface is damaged. So they have to take in high concentrations of oxygen so that whatever air gets to the surface, the concentration of oxygen is high enough that it can diffuse into the blood, right? And we have, how does it occur? Well, that's a little explanation there, right? The mucus, the bacteria, remember that accumulated mucus and bacteria that wasn't getting swept out of the lungs? Well, it stays there and it eventually falls down to the alveoli itself. And the bacteria is going to attract phagocytes. And the phagocytes, in their enthusiasm, is going to eat through the alveolar membranes to get to the bacteria. They're not checking to see what's going on. They're like, bacteria, run after it, bust through whatever alveoli, say, I don't care, I need to get to the bacteria. And therefore, you literally have an autoimmune response where, well, not autoimmune response, but a version of it where your immune system is now destroying your own tissues in its function, right? And the bacteria goes after the phagocytes, eats through the alveolar membranes, and of course, that's going to weaken them and they're going to eventually collapse. Because remember, these alveolar membranes are very thin. They're one cell thick. So if they're constantly having phagocytes busting through all the time, making teensy-weensy holes to get to the bacteria, eventually it's going to fall apart. Yeah? So that's how emphysema eventually develops in long-term smokers in particular, right? And that's question 4. As you can see, question 4 only had 2 parts, right? But the amount that you had to write for each one is a lot higher, right? So let's get to question 5 now. So question 5. Well, part A has several parts. So we went from a question with 2 parts to a question with parts of parts. So in part A1, we're being told, we're being, reminded that blood that's oxygenated, has to be pumped through the heart before it's sent to other organs. And you now have to describe the pathway. So question 4 was dealing primarily with respiration and the respiratory system. And now we've shifted to the circulatory system. And just an aside, this topic, circulatory system, is a topic that seems to be problematic for candidates across the region. So if you are able to handle a question like this well, you will rank quite highly. It's going to definitely aid you in your journey towards grade 1. Yeah. And we have here, so we're basically being asked to describe your cardiac cycle, essentially, right? The flow of blood. And I shouldn't say cardiac cycle, that has to do more with the heart, how the heart, the coordination of the heart pumping. But essentially the flow of blood through the heart as it pumps, right? So let's take a look at, and they're asking you to start from the lungs. So note that you want to take note where you start. So you're starting from the lungs, flowing from the lungs, and then sent to the rest of the body. Okay, so already you're going to be looking at your what vein is bringing blood to the heart, and then we go on from there, right? So the first step, the blood enters the heart from the lungs via the pulmonary vein into the left atrium, right? And note that we're making sure to name the vessel, and we're naming the chamber, right? Then the left atrium is going to pump that blood into the left ventricle past the bicuspid valves. This should be your tricuspid, right? And then the left ventricle pumps the blood out of the heart via the aorta. The aorta connects to other arteries, transport blood to the body, and the valves prevent backflow into the left atrium, right? And that's basically it. Notice that you're not being asked to describe the entire flow. So you don't have to talk about blood coming from the body into the right atrium and all of that. You're just talking about one half of the cycle, right? Which would what's considered your, remember you have two circulations that occurs because we have a double circulatory system. You have your pulmonary and your systemic. I know some people I remember a student asking, is this systemic or systematic? Like, no. Systematic means something according to a system, something that is in a very ordered way, etc. Systemic means system, as in your body systems, right? Whatever the whatever Greek and Latin, wherever that came from. But basically, you're looking at the system, focused on the system, and that's systemic, right? Systematic has a different meaning altogether, right? And and that's it. Let's see, what else? I did include 1, 2, 3, 4. I did include the valves because that is also an important part of the function, because if that doesn't work, the blood is not going to flow out to the body through the aorta as it should, at least not as efficiently as it should. So those valves are very important to make sure that all the blood that's pumped out of the ventricle actually goes through the aorta and out to the rest of the body, right? So that is an important feature. And that's your 4 marks, right? Then we have the next part, physiological diseases affecting the circulatory system, right? 1, 2, 3, hypertension, atherosclerosis, coronary heart disease. Those are your physiological diseases. Right, so we have here part B, and we're being asked to explain why chemotherapy has an impact on natural immunity. And the opener described the fact, or provided the fact, that chemotherapy destroys both bad cells and good cells. So that also gives us a hint that we want to focus on cells, right? So we're looking at how the destruction of good cells has an impact on natural immunity. And as per usual, you want to start by describing what natural immunity is, and you also want to describe what natural immunity is with respect to the cells. All right. So here we talked about how you get natural immunity to become immune to some particular disease or pathogen or toxin, right? And you basically you get natural immunity by from prior exposure, and antibodies are developed, and you now have memory cells, important, that can then trigger a more rapid and more intense response on future exposure. And that's essentially what natural immunity is, right? And then we talk about how chemotherapy affects that. So chemotherapy would destroy many of those lymphocytes, especially the memory cells, and that would then destroy your immune system's capacity to deal with these pathogens, and hence your natural immunity is gone. Because remember, a lot of your natural immunity is dependent on your lymphocytes, and they're dependent on these memory cells that stick around in your system, so that if the same pathogen shows up again, boom, deals with it quickly and intensely. So the chemotherapy now killing out, destroying all of these cells, right? And then you also have other types of cells, right, that are also part of your immune system, part of your immunity, right? Things like your phagocytes, which are going to go around eating up the these pathogens. And in fact, if we remember, not pathogens, phagocytes, they team up with lymphocytes, because what some of these antibodies do is that they cause the pathogens to clump together, and that makes it easier for the phagocytes now to come in and consume them. So the phagocytes being destroyed also is going to have an impact on your natural immunity, because they are like the mop-up crew. They will come in and deal with those pathogens, right? And then you have your epithelial cells, all this, those, any type of cell that is a lining that covers the lining of your body. Remember, those are entry points for pathogens. So you have your skin, which will be a distinct barrier against entry of pathogens, and then you have your various openings like your nostrils, etc. You would have epithelial cells that will produce mucus to trap the bacteria. You have epithelial cells that will produce acid in the stomach that also helps to destroy certain types of pathogens once they come into your body through your mouth with you eating foods and so on, right? So if those will be affected as well, all right, they may not be destroyed as, the epithelial, because they're able to regenerate so quickly. They're not utterly destroyed, right? But at the same time, they are significantly affected, right? Well, the phagocytes definitely is going to be affected significantly as well, right? So that's basically how you can answer a question like this. Then we have Peter, who has not had a tetanus vaccine. He got his foot punctured and so on. So they're asking, why was he given anti-serum instead of a vaccine? So first you need to indicate how the vaccine works, right? It works by triggering the primary immune response. That's what your specific immunity has to do with, right? That's where you are getting the lymphocytes to produce antibodies or antitoxins against whether it's a pathogen or a toxin. But that takes a while to occur, right? It takes a while for the lymphocytes to manufacture the right antibody or antitoxin. And if you already have an infection, or you already are exposed to a particular toxin, you can't wait for the lymphocyte to go and produce the antitoxin or antibody. So in that case, you give them the anti-serum one time. So an anti-serum would be like giving the person ready-made antibodies or antitoxins. So as we see here, tetanus acts faster than the primary immune response could work. So therefore, giving this ready-made antiserum, it's going to work quickly enough. It's going to neutralize any toxins that are being produced to protect the young man, right? Then we have our last question now, where we're being asked to describe how genetic engineering is used to produce human insulin using the bacterium E. coli. And 6 points. So 6 marks, 6 points, 6 steps. But you can break it up into 6 steps so that you can keep track of the marks. And that's a little tip again. If you have a question that has a lot of marks in them, break up your answer into parts, whether you can make a numbered list or a bullet list, or you break it up into small paragraphs. You basically organize your answer. Okay, so basically I'm looking for 9 points. If it's 9 marks, I'm looking for 9 points. What are my 9 points? How can I organize my 9 points? And then write it out. Don't just write stream of consciousness on the paper and hope that the points emerge. Now, the examiner is going to read all of it, and they are going to look for those points to give you the marks. But if an examiner comes across an answer that's well organized and all the points are placed out really nicely, then they're going to really appreciate it. They're you will get those marks a lot easier because it'll be easier for them to read what you're putting forward. And then it's easier for them to then apportion the marks that are needed versus them having to read your stream of consciousness 4 or 5 times to find 3, 4 points, right? So as we said here, 6 points, 6 steps. And in this case, you can use a numbered list. I'm just using my bullets, right? So first point, you want to use a section of DNA that codes for human insulin because you want to find the gene for human insulin. That's what a gene is, a section of DNA that codes for a particular protein. And in this case, the protein is insulin, right? And yes, by the way, insulin is a protein. It's a very simple protein, actually. And this is going to be either extracted from human cells, or you can synthesize them from pancreas tissue using reverse transcriptase. Reverse transcriptase is an enzyme. And just a quick explanation of what's happening there. A reverse transcriptase enzyme is going to take RNA and produce DNA using RNA as the template. It's an enzyme that you find in certain bacteria. No, it does not exist in humans. It's not in human cells. It's exclusively found in bacterial cells. And in pancreas tissue, remember pancreas tissue is where insulin is being produced constantly. And just a quick aside as to how the protein synthesis works. RNA is used as a temporary, it's used as a template, a temporary template for various mechanisms, various organelles, your ribosomes, etc. is going to use that RNA as a temporary template to manufacture the protein. So if you have a cell that's producing a lot of a particular protein, then you're going to find a lot of RNA for that protein present in the cells. So if I go into pancreas tissue and I extract the RNA, a large portion of that RNA is going to be for insulin. And so once I can isolate that RNA, I can then use the reverse transcriptase enzyme to take that RNA and then produce a DNA strand based on the RNA, right? So that's why you can take pancreas tissue, pancreas cells, use reverse transcriptase and produce the DNA that you want. right? And this DNA strand is going to be added to plasmids from E. coli bacteria. If you remember plasmids, if this is the bacteria, you have your nucleoid, right? Your large loop of DNA, and then you find these cute type loops of DNA floating around the bacteria. These are your plasmids. So what I can do is take a set of E. coli, extract these plasmids from the E. coli bacteria, and then I'm going to use these plasmids and via various processes, I'm going to now add this DNA strand to the plasmids. And these modified plasmids are now going to be inserted into other E. coli bacteria. So now you're going to be adding modified plasmids into the bacteria. And these plasmids are going to add the insulin DNA to the bacteria's genome, right? It's now floating around in the bacteria. And therefore, the bacteria is going to treat it as one of its own, and it's going to read the DNA for insulin and manufacture DNA, manufacture insulin, right? Because that's the DNA. DNA says make insulin. Okay, I'll make insulin. And it's that, I don't want to say simple, but it's that straightforward, right? And of course, there are lots of complicated steps, but that's essentially what's happening there. And as the bacteria multiplies, you place the bacteria in a fermenter. As the bacteria multiplies, the modified E. coli, they will be producing insulin, right? So they will multiply and they'll make lots and lots and lots of insulin. And then after a certain point, the insulin is going to be extracted from the fermenter, right? Just like when you go into the carrot factory and they have these huge fermenters and they're producing, they're going to use yeast, right? So they're using yeast organisms to produce lots and lots of alcohol. And then after a while, they're going to extract the alcohol out of the fermenter. Same thing, I shouldn't say same thing, but a similar thing is happening with the production of insulin. You put it in a fermenter, let it do its thing, it produces the insulin, and after, you will extract the insulin from it and purify it, put it in little bottles, and sell it to people, right? So those are your answers there. Then we have 3 differences between genetic engineering and artificial selection. And when we talk about artificial selection used by farmers, we're talking about things like breeding, right? Right? When you're breeding different types of pigs and cows and horses, all of that would fall under artificial selection. Now, note that genetic engineering is a type of artificial selection. Well, actually, no, I shouldn't say that because no, it's not. It's what you would call, is it a type of artificial selection? I think that one is up for debate under certain definitions, yes, under certain definitions, no. So in this context, we treat it as a separate. It is a type of biotechnology. That's definitely what it is, right? Biotechnology, right? And artificial selection would include like your breeding, like, so for example, horticulture. Horticulture involves a lot of artificial selection, yeah? And there you go. So let's take a look at the differences between the two, because in fact, here, we're talking about crop plants. So we're not even talking about cows and pigs and whatever. All right. So let's see what the options are. Now you have to choose 3. Notice you have a lot to choose from until I had to make like a table, right? Doesn't even fit the screen. And notice it's 2 marks per point, meaning you can't just talk about one part. So for example, if I said, Well, artificial selection, you're going to get a mixture of desired and undesired traits, full stop. Then that's 1 mark, if at all. Right? Some cases, they might even give you the mark. You have to talk about the both sides of the argument. So yes, for artificial selection, you're going to end up with desired and undesired traits because you can't really control that. Whereas with genetic engineering, you can target only the desired traits, right? So that is 1 point of comparison. And as we can see, we have lots of others. And I'm not going to use up your time going into, right? But there is a lot of answers here. Now, as I said before, the ebook, this same ebook that we're looking at right now will be available for free very soon. Yes, long before the actual exam. Those of us that are watching this in 2023, it will be available before your exam on the 22nd. However, what I would need you to do is go to the link in the description below. That's what they call it, description. So you go into the link in the description below and you pre-order it. And as soon as it's available, I will then email it to you. I'm not asking for much information, just a name, how you call yourself, and an email, right? And then we'll see what happens from there. Once I get your email address, as soon as it's available, which will, again, long before your exam, I will then just blast it out to everyone, right? So as you can see, I even as I'm doing this, I'm making adjustments as well. So the version you will get will be updated, right? So yeah, because we're not gonna go through all this now. But again, and as I tell my students, when you have questions like this or topics like this that includes a lot of details like this, it's always good. Don't try to stress yourself out and try to memorize everything. Choose about 3 to 4 main points that really stands out to you, that really is easy for you to remember because you could connect it to something else and in your mind, and use that, right? Memorize that, because to try to memorize all of this, no, right? This is just on the examiner's end, trying to cover all possible bases so that once the person gives that answer, they can get the mark, right? Yeah, and that's the best approach with these kinds of things. For example, when you're looking at the sources of micronutrients, you know the list is long. So you remember 2 to 3 foods that will stand out to you, and you memorize those and move on. All right. Right. You don't want to be wasting your life memorizing a whole lot of stuff that you don't need to. And then we have the next question. Don't worry, the actual ebook will be a little more organized. But this question is asking you to suggest 3 advantages of using genetic engineering to produce insulin, as opposed to, which was the before, they used to extract it from pigs and cows and use that to treat patients. So firstly, well, not firstly, but it's one of the top points that I started with, is that genetically modified bacteria, you get to produce insulin in a shorter time, which is important. Because you don't want to be spending more than the necessary time to produce the insulin, because there's a lot of people that are out there waiting for that insulin, yeah? Then you get to produce a lot more insulin. The amount of pancreases that you have to extract from pigs and cows in order to then remove insulin from it, extract insulin from it, it's a lot. You're talking about tons to get a few grams of insulin. Versus you take some bacteria, add the DNA to it, put it in a fermenter, just like in the carrot factory, and you ferment, and then you extract. And the amount of insulin that you get from that is so much more. All right. And again, because you're producing a lot more at relatively lower cost, it is cheaper for the consumer, right? And it should be a lot. In fact, it should be a lot cheaper than it is right now. But we can thank corporate greed for that, for it being a lot more expensive than it needs to be, right? But left up to just the cost of producing it, insulin should be dirt cheap, right? And the fact that the person who invented it, came up with the way to produce it, he didn't even add a patent to it because he wanted it to be freely and cheaply available to the population, right? Because he wanted to help, diabetics, people who are diabetic, who are suffering from this disease, right? So, but that's corporate greed for you. Yes, that's my little soapbox moment, right? And then we have humans do not develop resistance. That's true, because it's human insulin. So our bodies are not going to reject it or resist it, right? And then that brings us to the next point where we don't have as much allergic responses. And the other part, which people don't really think about as much, is that if someone is Hindu or Muslim, they may have a problem with accessing insulin that's made in other ways. Why? Because they come from pigs and cows. So that, makes the genetically engineered insulin maybe more acceptable. I say maybe because I know people still do have a resistance to the idea of genetic engineering, but at least on that religious point, it removes that problem in that I don't have to worry about taking this thing into my body that comes from a pig or from a cow, right? So that's another advantage. And that's it for paper 2, January 2017. Yeah, that's it for that.